In this video, topic 7.8, we'll be examining continuing evolution. So if you remember from earlier videos, we talked about the agents of evolutionary change. These include changes in the DNA, which we call mutation, gene flow, which is the immigration or emigration of traits within a population, genetic drift, which is chance events, and lastly, natural selection, selective pressures where one of the traits has a selective advantage over other traits within the population. So how do we see that evolution continues in our populations today? Well, one of these examples is the beak length of the finches on the Galapagos Islands. So as you can see in my diagram right here, in about 1975 they started measuring the average beak depth of the birds. And from 1975 all the way to 2010, we see that there's a change in the average of the beak depths. These changes correlate to either dry seasons or wet seasons in the islands. And what that means is that there's a change in the food source. So certain birds have advantages based upon the shape and size of their beaks. If that food source is more prevalent, they're going to see a shift in the beak size in that direction. So this is an example of our population changing over a very short period of time. I want you just to remember that evolution doesn't take millions of years. It can actually happen very quickly. Another example of this idea of continuing evolution involves the genomic changes over time. Genomic changes involve random mutations that introduce variation into the population. Remember that what we're looking at here is we're looking at mutations of the DNA, and a lot of the times these mutations are random. We don't know where they're going to happen. Sometimes the change is beneficial, sometimes it's detrimental, and sometimes it's neutral. But remember, only the beneficial changes are going to be passed along into future generations. You can see right there in the bottom left-hand corner really what these random mutations may occur. So if we randomly mutate our DNA, it may by chance happen to change the color of the fur in the mouse, going from the white color fur to the black color. And the black actually may be an advantage or it may be a disadvantage. And as you can see there on the right hand side, this is just the example of the changes in the bacteria population. So when there is a mutation as represented by the lightning bolt there, you can see that some of these mutations are actually beneficial, where some are detrimental, such as that red one there. That mutation in the DNA actually caused that particular strain to die off, where it seems as if the blue species, which resulted in actually a second mutation, has the selective advantage. So following this from generation to generation, we can see a population evolve. Another example of how species can evolve or continue to evolve is if we look at the fossil records. Now, in an earlier presentation, I talked to you about this peculiar observation of pelvic bones in a whale. So why would a whale need pelvic bones? Well, that is an example of a vestigial structure. And based upon the fossil records, what scientists believe now is that the whale was once a creature that walked on land. And through millions of years of evolutionary selection, you can see how that whale that was once a land-dwelling creature lost its legs and became more streamlined for the aquatic environment it lives in today. So looking at the fossil record allowed us to see how this animal evolved through evolutionary time. Another example of this continuing evolution is the evolution of resistance to antibiotics, pesticides, herbicides, or chemotherapy drugs. Now, in the last few presentations, I've talked about how antibiotics bacteria become resistant to antibiotics, and that's shown there in the top of the slide. So rather than dwelling on that example, I'm going to talk about the example of a cancer that becomes resistant to chemotherapeutic drugs. So you can see right there in our tumor is represented by the white cells and the blue cells. By random chance, the blue cells have a genetic mutation that make them resistant to the drugs. When the tumor is treated with the chemotherapy, notice that the vast majority of the cells regress, die, disappear. And the chemotherapy drug targets the susceptible white cells. However, what you'll notice there in the middle of the slide is the blue cells are resistant. And over many generations or many treatments of the drug, what you'll notice is that this tumor has now reoccurred. That's because the drug killed all the sensitive tumor cells, where by random chance, the ones that are resistant to the chemotherapy drug now are prevalent in the tumor population. And the last example here talks about pathogens and how pathogens evolve, and because they evolve, they can cause the emergence of new diseases. Now, one of the examples of this is actually influenza. So influenza has 9 genes, and what interestingly with influenza, we all know that we have to get a new flu shot every year. That's because from year after year after year, the virus actually evolves. It changes. The genetics of it change. And the vaccine that you got last year may or may not be able to fight off the flu the next season. That's because when flu, we can have what we call a genetic drift and a genetic shift. And basically what happens here is the virus mutates so that our immune system that once recognized it as a foreign pathogen and was able to eliminate it, maybe the following year or the year after that, there is a genetic change, and now the virus has evolved to evade our immune system.